Thank you very much for the very kind introduction, and thank you very much for the organizers to allowing me to be present at this really exciting event. So, I was assigned the task to talk about spatial biology, but as I am a pathologist, I take a biopsy-centered approach and a very practical approach, because my main disclosure, actually not on this slide, is I'm absolutely not an expert in spatial biology. But I am an end user at the lowest competency level. So when I can use this technology and it makes sense to me, we could probably all use it in the clinical context to improve patient care. So technically, I'm the canary in the coal mine, usually in a lab. When there is a new machine or assay, they let me play with it. And when it survives it, they say, okay, it's good enough. So why the biopsy? It's one of my mentors, Phil Halloran, said it's Sutton's law. So everybody knows Willie Sutton in the room. He was a notorious bank robber. I think he robbed over 60 banks. And when he got caught, he was asked, why do you rob banks? And he says, that's where the money is. So the biopsy is where the truth is, where the disease is, and that is what the grandfathers of pathology established with Robert Koch at Koch's postulate. You have to see where the disease operates, and you have to see the cause of what causes the pathology to understand and treat it. Today, this is called precision medicine, but it's a 100-year, 150-year-old concept. And I'm just wondering, did all our mentors and the people we trained with do non-precise medicine before we started doing precision medicine? So Robert Virchow is a German pathologist who established this concept that the pathology starts at the cellular level. And his trainees linked this to Koch's postulate. When you have an infection, you get neutrophils, and the neutrophils, together with the cell debris of the inflammatory reaction to the infection, causes the pus. At that level, people know you have an infection when this big pus thing blew up, right? And it smelled and it all came out. But it was revolutionary taking it from that macroscopic level to the cellular level. And I guess the topic today is to take this a couple of levels further. Still, we are dealing with an inflammatory response. In this case, the pathogen, according to Koch's postulate, is the transplant, which causes the inflammatory response, and we try to understand this. And I do not need to educate this audience at the 101 level, but I need to remind myself we're walking a fine line in organ transplantation between a natural immune response and suppressing or manipulating that with very sophisticated tools to avoid rejection at a certain cost. We're also potentially suppressing regular immunity, and we're buying infection. And our response to infection looks very similar at the pathological level to rejection. And something we struggle with, classical example of T-cell mediated rejection and BK nephropathy, which is almost indistinguishable by light microscopy in the biopsy. And I'm focusing now more on kidney transplant pathology, because the kidney is the highlighted example of morphological compartments. And the whole pathological assessment of a kidney biopsy is surrounded by, surrounds these compartments, these morphological structures we see in the organs. And that is the foundation for the current diagnostic standard, the Banff classification. As you see in this picture here at the bottom left is a tubulus with tubulitis, the hallmark of rejection for the last 30 years. Or on the right for antibody-mediated rejection, an example of a glomerulus with microcirculation inflammation. As you heard in Dr. Buddha's talk, the main target of the most fanciest recent drug to reduce that pathology. So we still use these fundamental concepts of monitoring response to treatment, of diagnosing it into categories of rejection, and to guide patient management. And on the left top, you have the over 30-year-old Banff concept of these compartments, associated pathology, mainly inflammatory features, and a semi-quantitative consensus-based scoring system. 1991 in Banff, Alberta, people came together and said well, if there is more than 25% of interstitial inflammation, we call it rejection. At that time, it could have been 20, could have been 26. It was just a good number. But what it formed was the foundation to titrate immunosuppression to get inflammation below this threshold in the graft, which was strongly associated to improve 1-year graft survival, I think from 30% to almost 80% over a period of 5 years of clinical trials after the introduction of calcineurin inhibitors and MMF. Fundamental, but we are still at that point bringing the canonical lesions in pathology just below a threshold. And all these lesions, and this is now the dirty secret of transplant pathology, are absolutely not specific. You can see them in various different disease contexts. So it is contextual. And then, again, Phil Halloran said, Michael, sell your microscope. We have microarrays now. You're out of business. I'm still here. Okay, I'm tenured. It's a bit of an unfair approach, right? They can't fire me. But I'm still using the microscope. And when I came to Canada in 2006, right, there was a big Genome Canada grant. A lot of microarray studies done since then, and I will summarize those results in the following. Pivotal paper by the Halloran Group, Thomas Müller, now retired in Switzerland. I envy him. The fundamental finding on the left top is when you study all human genes expressed, 25,000 roughly, in a kidney transplant biopsy, you see a fundamental relationship between more T-cell inflammation associated transcript, blue line at the top going up, gray and black lines at the bottom, regular metabolism in tubules and nephrons going down. Summary, inflammation bad, right? With Virchow and Koch kind of said. And that correlates with Banff lesions at the very top. Red means high Banff scores, right? Orange, medium, on yellow, and black, 0 Banff scores. So the more I see in the microscope, the more you see in a microarray of the same thing. Fundamental really, I'm oversimplifying, okay? There's a lot of information there. Interesting on the right is take the top 10 transcripts of T-cell associated inflammation, CXCL11, 12, 13, gamma interferon associated transcripts, granzyme B, pick your favorite. They all correlate by 0.9 in these data. They all tell you the same fundamental relationship when you measure them. There's not a single molecule expressed in a transcript after more than 10,000 microarrays done, which is specific for rejection, and which cannot be upregulated under different circumstances. The molecules are as specific as the histology lesions in the Banff classification. There, you can measure them more accurate. They're more sensitive, they can be quantified, it can be reproduced. They have a lot of analytical advantages, right? Don't get me wrong in that. However, all these studies were done as bulk expressions. So you get a chunk of tissue, you grind it up, you extract all the RNA or DNA or whatever your favorite, all peptides, and you do not have the compartment or spatial context, which histology has. And we know that the spatial distribution of certain pathologies changes over time post-transplantation. So this is a study we published back then. The longer you're out after transplantation, the more scarring you have, the less nephrons you have. different types of inflammation come up, more mast cells, more B cells. So you establish a chronic inflammatory compartment in your allograft, as you do when you have an abscess, right? When you had an infection and it doesn't really heal, you build around scarring, you get more chronic local inflammatory regulation, and the same happens in the kidney. And the Banff lesion scores kind of reflect that. We score now inflammation in areas of fibrosis, the IFTA score. We know that is one of the strongest predictors of outcome because it shows activity and ongoing injury to a graft and is, so to say, subacuity compared to the very early rejection pictures. That's not necessarily priori reflected in bulk expression, but when you train classifiers against histology, you can tease it out and give you the information of that as well. And again, we did molecular studies of PD-1 inhibitor-induced interstitial nephritis in transplants versus drug-induced interstitial nephritis versus rejection versus BK. And unfortunately, the molecules are heavily overlapping. The most discriminatory gene in BK nephritis is the BK gene. Duh. The inflammation is almost identical at the molecular level. So what this paper really by Phil Halloran summarizes up of thousands of microarray studies. We know which genes are most identified, or we identified, the genes which are most associated with certain pathology phenotypes. So when you have antibody-mediated rejection, the top transcripts are endothelial transcripts and K-cell transcripts, very robustly. Whatever method you apply, whatever study, it's a fundamental relationship. T-cell mediated rejection, it's gamma interferon inducible chemokines, it's cytotoxic T-cell associated molecules, and it is a downregulation in the context of rejection of functional activity of the kidney or your organ. When you're in the heart, the myocyte activities, glucose transporters go down like in heart failure. Or here in acute kidney failure, it's glucose transporters, it's soluble transporters, so your nephrons lose functionality. That's why your creatinine goes up eventually, right? It all makes sense. Very fundamental relationships, and when you have cases with mixed rejection, those genes overlap, and the degree of injury and the quantitative burden of inflammation tells you what your prognosis, so to say, is. So these are very fundamental Koch-like relationships at the molecular level. So what Banff then did is, in the second era, so to say, of Banff, 30 years, these fundamental relationships were reflected in the Banff lesions. T-score, I-score, G-score, V-score, fundamentally. And we tried to do the same for the molecular relationships. Genes associated with inflammation, injury, infection across the various organs, the top transcripts in all these studies, we filtered out and put it into one panel. The panel is commercially available by NanoString, but you can download the panel list and create it on any other platform. So the genes are not proprietary. But those are, so to say, the consensus molecular phenotype and lesions in an allograft biopsy. And you can run it on whatever platform you wish. You could run a microarray and filter out those 770 genes. And as Banff is, we don't claim they are perfect. Based on today's knowledge, they are the strongest associations we have. And now people apply this, because it's out there, you can use it with whatever technology, as I said, you want, and start putting it into the spatial context. And you find these associations that microcirculation inflammation has a higher NK cell signature, as Ivy Rosales, for example, published in JASN for peritubular capillaritis and chronic active antibody-mediated rejection. But what Ivy did, and she is probably one of the leaders at MGH in spatial transcriptomics in antibody-mediated rejection, studying nieces. She was able to link this back to the classical morphology. You see electron microscopy at the bottom, where you know that NK cells interact with the endothelium. But she then looked at the counterpart of the NK cells, what endothelial transcripts are changed in this context. So you can discover fundamental mechanisms and molecules like EHD3, which is a membrane remodeling pathway, generic in many injured cells, but that precedes the duplication of the glomerular basement membrane. So the inflammation injures the endothelium, the basement membrane tries to heal, and that's when we start seeing transplant glomerulopathy, which is the strongest predictor, negative predictor in chronic active antibody-mediated rejection. So this technology starts to give you mechanistic insights at the spatial direct interrelationship. And you can spend the rest of your career as a postdoc on one of these two cells with two molecules. It's great. Other technology for single cell sequencing around finding and confirming, Ben Humphrey's group early on published in JSON, these fundamental relationships I showed you from Thomas Müller's paper. Inflammation goes up, tubular function goes down, microcirculation inflammation in peritubular capillaries associated with endothelial degeneration. These spatial studies confirmed these fundamental relationships, which are linking back to fundamental pathology. Alex Lupi and other consortia now applies this to the xenotransplant and the Decene models, which gives you a controlled environment to study all layers I just discussed. Very nice paper here to read, where he goes from gross pathology, light microscopy, protein expression with immunohistochemistry, to BHART expression, and then single cell sequencing at the spatial context. So bulk RNA, and links it down and shows and confirms these fundamental relationships we're finding. And you can now, in these xeno models, do this over time in sequential biopsies under controlled conditions. And that really opens up when to do the intervention, because usually James or somebody else does the biopsy when the patient already deteriorated and already has transplant glomerulopathy. But we will now know what are the earliest signs of this pathway, and that can tell you when to do the targeted drug intervention, like with CD38. Same applies for recently just published in the heart. I think there's another paper coming around the heart xenografts showing, again, the fundamental relationships between cellular function, endothelial function, and microcirculation inflammation. So the fundamental antibody-mediated rejection pattern at the molecular level is unfortunately found in all these xenografts. So we thought we solved it by manipulating 11 genes, but it's not resolved yet. And this is a recently published in AJT published paper by the Leuven group with Martin Nasons, and he focused on the tubular expression of the nephrons and could show at the single cell level that the fundamental relationship Thomas Müller published, that the solid carriers, the glucose transporters go down when you have interstitial inflammation, and when you have microcirculation injury in peritubular capillaries, are downregulated. So the nephrons are very dependent on peritubular capillaries in their functionality. And that's the next target. We focus on the glomeruli for transplant glomerulopathy, but how do we preserve nephrons as a major function in the kidney graft? So in summary, so far, these technologies confirmed at the cellular and spatial level these fundamental relationships we assumed. Even 30 years ago, saying, okay, interstitial infiltrate and tubulitis, that's not good for the nephron. But here we now know which molecules are involved in this and when it actually happens in sequential biopsies of these recent xeno models. So it raises more questions, and this is where we'll close is. And I gave a similar talk, and then Dori Segev, some of you might know, and he got up to the microphone and said, Michael, so what? What are we going to do with this? The question, what I'm interested in, so there's a lot of mechanistical insights, don't get me wrong, right? That might be druggable target. But as we talked about epitopes and which are real and not real and what all the possible iterations are, there is millions of iterations of molecule and cell-cell interactions in this. I just can't handle it, and therefore somebody else can find the right druggable target and not me. But I am wondering really about spatial technologies is, can we banfinize this and essentially get a molecular tubulitis score? A preset panel of genes which measures all the genes in 100 tubules, like I do looking for counting lymphocytes for tubulitis. But it gives you a quantitative score of X CXCL12 in that space with X burden of granzyme B and X downregulation of solid transporter 16. And that's a molecular tubulitis score of 3. And you could do this automated. You just load the slide, it gets scanned, and you get those numbers. And maybe that tells us better acuity, which drug you use to manage that acute incident. Is it a gamma interferon blocker for now, just steroids? Or is there more going on in the microcirculation injury already that we want to switch to a different drug? So I think the vision for this technology from an end user perspective is using it as a molecular BAMF scoring machine, so to say, and producing the light microscopic picture and the molecular level in the spatial context of a kidney transplant, for example. So I think that is what and I heard you can combine now these platforms with mRNA measurements, protein measurements all in one, so I don't have to score C4D anymore. It just gets counted automatically, or the copy numbers of that get counted in the context of C4D protein burden versus mRNA endothelial injury burden gives us better diagnostic precision. With that, I just made it more complicated, but thank you very much for the opportunity, and I'm happy to take any questions.
Thank you very much, Professor Mengel. We have a couple minutes for questions. I'll start with you, Paul, and then Dimitri.
Thank you very much. That was great. We now have the possibility to test all your hypotheses, because this will be incorporated in the gap, the epitope matching going forward. But it makes it much more complex, as you say, but it raises one particular question. This can be either an endpoint for the question, or it can be a guide to change in therapy, or both. And I was thinking as you were talking, we're seeing the risk of rejection fall, clinical rejection, to vanishingly small levels. We don't tend to do routine biopsies, but you have to do that if you want to do this.
Well, cell-free DNA is pretty sensitive, probably too sensitive, right?
Yeah.
Under today's, but I actually think that cell-free DNA, if the prices come down, will trigger more biopsies in just other patients, right? And we define arbitrary protocol biopsy by saying your creatinine is stable, and there is not even a definition of what a stable creatinine is. Is it 10% change, 20, 30, right? It's all over the map. So I think with cell-free DNA or other minimal invasive markers, you can find the at-risk population and focus on those. And the other aspect of the molecules is they are way more sensitive early on for those people who show early signs of endothelial injury. Yeah. But they are also way more sensitive in showing a response to treatment. So we don't know what that means in the clinical context. Is it good thing, or is it overly sensitive or not? But those would be, to me, the first questions to find out that when you do an intervention, how does the molecular response align with clinical outcome?
Yeah, that's great, thank you. in practical terms, we've already started talking to Mei-Lin and the team here about digitizing the information, and you'd kindly agreed to sort of be the coordinator of it. But we need now to begin to work on the actual mechanics of it, because I think we'll be launching into the study very soon. It actually gives us a wonderfully exciting opportunity.
No, I agree. Right. Currently, the spatial technologies are very cumbersome, expensive, and discovery tools. But I think the technology will mature, as always, that you can use it at a larger scale of biopsies and pair it with your clinical trials you're designing, right, to learn about these endpoints. I agree.
Mike, thank you very much for your inspiring presentation. I want to ask you to dream for a minute. If you were to need another technology to progress in your search, what would that technology be? We move from the microscope to RNA microarrays. Now we break down the tissue into cells and find out signatures of changes. Tell me, what would you like to have in your armamentarium to solve your problem?
Well, I'm a humble person. I think the computational biology is the next tool, because you generate so many data and the risk of finding accidental irrelevant associations is too high. So what is actually relevant probably goes beyond looking through a spreadsheet and finding a pattern. This is how I started with these microarray data. We looked through every row and said, oh, this gene sounds familiar. And it comes up everywhere in every case. And then we started a supervised analysis for a certain gene. That's not possible anymore. So, and what I struggle with all these AI pipelines now in lab and in prediction models is, how do you quality control that? Because they find something, it's like ChatGPT. You can put in whatever into ChatGPT and it comes up with an answer. And when you like the answer, then you say, oh, ChatGPT is right. But we don't even know whether it's true. It could be completely confabulated through AI. So I think the biggest challenge is really how to develop valid or validate AI tools helping us putting all this information into the right clinical context.
There's an interesting theory in physics, complexity theory, which is bordering to chaos theory. But they can identify patterns there that are very interesting out of a very complex set of data. I can talk to you because we used it. And we have beautiful results regarding the organization of the DNA sequencing in the whole genome. There is logic in every single part and pattern, whether it is exon, intron, or intergenic regions. And you can see the patterns there.
Well, when you talk to evolution biologists, right, they say that all these immune responses, replication of DNA, right, cell cycle, has a component of chaos, but evolution selected hardwired processes which give you survival benefit at the species level. But you always produce versions which have disadvantages. So I'm convinced that there is chaos in the immune response. It's not always following one blueprint.
Right. I have to stop the discussion here.